So with this, I'll introduce the first speaker to you, and this is, unsurprisingly, it's Julie Suman from Apta Pharma. She's the Vice President of Scientific Affairs, and she holds a Bachelor of Pharmacy and also a PhD in Pharmaceutical Sciences. She's also the co-founder of Next Breath, and already in her doctoral research, she looked into the relationship between in vitro tests from nasal sprays and in vivo deposition. So ever since, she has been very active in the field of nasal drug delivery in both analytical and therapeutical aspects. So I think she's an excellent choice to open the session and introduce you into nasal drug delivery. So over to you, Julie.
Yes. Thank you, Regina. It's a pleasure to be here. I'm very excited that we have an extended session on nasal drug delivery, and I'm pleased to kick off this session. And hopefully I'll set the stage for the speakers that are going to come for the rest of the day. And so I've been tasked with providing an overview of the current opportunities in nasal drug delivery. And the DDL organizers asked me to take a step back and look at the history of nasal drug delivery. And so if we go back to, at least from a pharmaceutical standpoint, it really starts with oxymetazoline and xylometazoline in the 50s for over-the-counter use. But then we start to see, in the 60s and 70s, the corticosteroids, Dexacort, which is no longer on the market, and believe it or not, an intranasal peptide, DDAVP, which is desmopressin, for systemic administration, approved back in 1978. The 90s saw a flurry of more corticosteroids and antihistamines for locally acting indications. And then fast forwarding through the genericization of these corticosteroids, in the last couple of decades, we've seen a massive growth, at least from the nasal drug delivery standpoint, in systemic administration into the nasal cavity. And that's really looking at treating different things like migraines, opioid overdose, treatment-resistant depression. We saw the approval of FluMist, nasal vaccine for influenza, as well as some targeted deliveries. We had Trihija, which was approved. It was just fluticasone approved for nasal polyps, as well as other life-saving medications. And most recently, in the last few months, in 2024, we saw the approval of Neffy, which is an intranasal epinephrine formulation for anaphylaxis. And, it's been exciting approval. Hopefully it will be a meaningful treatment for patients, provide access through the nasal route. And Neffy, in its clinical trials, actually has demonstrated that it has an onset of action Tmax of 30 minutes, which is actually comparable to the intramuscular injections that it was compared to. So the two IM injections, for example, had a Tmax of 20 and 45 minutes. So showing that nasal drug delivery for systemic purposes can actually be comparable to an IM injection. In addition, the device platform, which is the Aptar unit dose, meets the strict requirements from FDA in terms of dose reproducibility. So when we think about systemic nasal drug delivery into the bloodstream, we're really targeting the blue region, that respiratory mucosa with the turbinates, which increase the turbinate areas, which increase the surface area in the nasal cavity. So that's, really what we're looking at. But I'm going to spend the most of my presentation talking about targeting different regions of the nasal cavity, such as the olfactory region, the upper region of the nasal cavity, and the NALT for vaccines. But, when you take a step back and think about why do we want to deliver drugs to the nose, we just talked about Neffy, with it being able to have a fairly rapid onset of action, and it's non-invasive. And that really enables use of this platform for emergent situations, as well as be able to be successfully administered by healthcare givers or relatives or in an emergent situation. The nasal cavity also bypasses hepatic first-pass metabolism, which means we might be able to deliver a lower dose. For example, nasal glucagon, which is a peptide, has a bioavailability of about 27%. And so it really, especially for, things like peptides, the nose presents an opportunity to deliver, therapeutics like biologic molecules into the nose compared to perhaps, an oral administration route. And the nose, it's fairly hardy in that it has a good tolerability to different formulations and different device platforms. And so, I mentioned nasal glucagon. There's no doubt that it's a life-saving treatment. And I wanted to perhaps share, a different way of looking at the value of intranasal administration. And that from a cost-benefit standpoint, nasal glucagon has actually demonstrated that it save about $1,000 per treatment compared to the commercially available injection kit. And the reason why is that the injection kit is a lyophilized powder of glucagon. So the user or the caregiver has to reconstitute the powder, draw it up to the syringe, and deliver the dose. And so what the study that I'm quoting here has found is that a number of patients actually have trouble or incorrectly reconstituting and preparing the injection. So they don't get the right dose, and that takes them to the emergency room. Where with nasal glucagon, they can actually, use it correctly, they can use it quickly, and prevent the need to seek additional care, resulting in that cost savings. So within Aptar, we think about now the evolution of nasal drug delivery and looking at different targets. And certainly through COVID, we saw a rapid increase in the nasal route for vaccination and delivery of antivirals, things such as monoclonal antibodies. And so we're really looking to target either the dendritic cells that line the nasal cavity or the null, which that red area circled in the back of the nasopharynx. And so we had to quickly adapt some of our nasal devices or develop new devices that could work with the existing manufacturing processes. So you can see the first two devices there on the left, the Bivax and the Lorvax. They're transfer devices, so we are able to use the existing, vials and draw up that dose to be administered to patients. There's also nasal powders that are being explored for nasal vaccination. We talked a little bit already about systemic nasal drug delivery, and there's multiple platforms, available in that space. We have multi-dose, we have preservative-free multi-dose. There are the unit dose and the bi-dose systems, as well as nasal powders. So a growing, range of device platforms, in the space for systemic nasal drug delivery. And some of these devices can also be used to target the CNS. Drug that can be absorbed into the systemic circulation can go from the blood and cross the blood-brain barrier into the brain. But we're going to spend a little bit of time talking about targeting the olfactory region for direct nose-to-brain transport. And here we have two device platforms. Within Aptar, we have a targeted device that we have developed, as well as we've also recently acquired the SIP-Nose platform. So nose to brain, no doubt it's been a controversial topic. And when we think about the nerves in the nose, I have shown here a picture. We're typically talking about the olfactory or the trigeminal nerves, but there are other nerves in the nasal cavity, and this picture doesn't even really do it justice of all the nerves. But by far the focus has been on the first two that I've mentioned. And we've been talking about nose-to-brain delivery for more than 40 years. We've had presentations that talked about drugs showing up in the cerebral spinal fluid or the CSF as long as 40 years ago. But today, there are still no randomized clinical trials that have shown proof of nose-to-brain transport in man. We've certainly had feasibility studies, we've had indirect measures, but there's still, a lot of activity to really prove that this transport happens in man. We can see it in animals, but the question is, does it really translate? So, one of the things that always sort of, perplexes me is that we can actually have transport of viruses, bacteria, and toxin from the nasal cavity, from the olfactory nerves into the brain. For example, it's been demonstrated that the SARS-CoV-2 virus can actually enter the brain via the olfactory region. And that's because those nerves really provide a direct connection to the CNS. So, why is it that we're having, this challenge with the drug delivery aspect? So we'll spend some time talking about that, but wanted to share some latest research that in trying to demonstrate how these pathways work. And this is a study that was done by Dr. Suzanne Kraft at Wake Forest. Suzanne has been conducting a phase 2 NIH-funded clinical study on intranasal insulin for cognitive impairment in patients with Alzheimer's and mild cognitive impairment. And one of her missions is to truly understand how drug is getting into the brain. So her first step was to perform a PET study with a radio-labeled insulin into vervet monkeys. And so she performed, her first step was a dosimetry study to be able to, use this marker for human use. But the results here that I'm showing you from the NHP study, and that she used the novel radio-labeled insulin in our Aptar CPS pump and delivered into the monkeys. You can see there, the dose was administered upright and the animals had to lay down for the subsequent scanning. But what the results have shown is that we do see uptake into the brain of the animals, direct nose-to-brain transport. So if you look at the graph, we have the activity from the PET scan versus different regions of the brain. And what this shows is that the insulin was taken up both by the olfactory and the trigeminal nerve. So the amygdala, for example, is more associated with the trigeminal nerve. So we're seeing transport along both routes. And so Suzanne has conducted this study in man, and hopefully those results will be available in Q1 or Q2 of next year. So please, watch the space. I think she's doing some really groundbreaking work. And then, we've seen some studies in man, and this is also an intranasal insulin study. This is where we're looking at a biomarker change in patients with MCI or Alzheimer's disease. In this study, 49 volunteers were either given a placebo or an intranasal insulin formulation. And the researchers were looking at a change in a biomarker called white matter hypersensitivity, the WHM. So in the graph here, the gray bars are the insulin and the white are the placebo. So what we want to see is actually a decrease in that biomarker. So that's actually the outcome that's related to cognitive improvements. And after 12 months in this study, you can see that in different regions of the brain, there is a biomarker improvement in different regions of the brain, again demonstrating that there's some potential nose-to-brain benefits in man. So how does, when we look at the pathways, let's talk a little bit more in detail. So I mentioned the olfactory and the trigeminal nerves. The olfactory nerve on the top of the above the superior turbinate, it has one of the interesting features there is that there's actually limited cilia in that region, and so there's less mucociliary clearance, which may lead to more, retention time, which may benefit for transport. The trigeminal nerve innervates lower into the turbinate regions, and it has some different features compared to the olfactory nerve, but it's also subject to mucociliary clearance. So maybe that might be more challenging for transport. So when we focus on the olfactory nerve and we talk about those transport mechanisms, there are intracellular and extracellular. So the figure here shows the two pathways. Intracellular, which is if you think of the red dots as the molecule going to that black neuron here, going along intracellular transport. It typically occurs by endocytosis and can transport into the olfactory bulb. But that's generally thought of how most bacteria and viruses are transported. The more relevant way for drug transport is thought to be the extracellular route. So if we look on the right-hand side of that image, this is just one example of paracellular transport where the drug is crossing between the cells, across the lamina propria, and into the subarachnoid space for distribution into the brain. But within that extracellular processes, there are different things that we need to consider. In addition to diffusion in across the lamina propria, there could be transport along the sheaths of the nerves. So you can see here that the neurons have different layers surrounding them, and so they're called, they have different names, but generally it's forming a sheath. The drug can transport along there. There could be receptor media or mediator or cell-to-cell mechanisms. There's also lymphatic processes, and then there's perivascular transport, where there are blood vessels in this region, and it's thought that that's the way drugs are actually transported throughout the CNS, through around the vasculature into the brain. Oops. Some other things that we need to think about, and some things that may actually work against us, is the efflux of CSF fluid from the brain into the nose. This is a couple studies that has demonstrated that route is possible. So on the right-hand side is a study in rats where they injected a blue dye into the back of the brain. We have a little cartoon image here on the back of the brain, and here's the nasal cavity. And if you look at B and D, so this is the nasal region of that rat, you can see that the blue dye is actually leaving the brain into the nasal cavity. So there's actually bulk flow of this CSF into the brain. Most research thinks that's along osmotic pressure gradients. And so it's been demonstrated in animals, and we're starting to see studies that are showing this in humans. So on the right-hand side are PET scans of adult volunteers. The top two rows are normal volunteers, and the lower level are patients with Alzheimer's. So if you can see the red voxels here, so the tracer was injected into IV, it went into the brain, and then you can see that it's being cleared into the nasal cavity in both normal and Alzheimer patients. So there is another pathway that we could potentially take advantage of. So when that CSF actually drains into the nose, there are lymphatic systems to remove the drug. So if you follow the little yellow lines here to the cervical lymph nodes, that's actually pathways of lymphatic drainage into the different various lymph nodes. And so that is also a potential pathway that we need to consider. And there's an example of this product currently, or this pathway being utilized in clinical trials right now. So intranasal formalumab, which is a monoclonal antibody with currently being investigated for multiple indications, but in this case, I'm going to talk about the progressive multiple sclerosis aspect. And so the hypothesis for the mechanism of formalumab is that the molecule interacts with the lymphoid tissue, and it results in a change in T cells in the cervical lymph nodes, and then which convert the T cells into T regulator cells. And that is those T regulator cells that are transported into the brain to exert their effect on the microglia. So another pathway for us to think about. And, so what does this mean to us, as drug delivery experts in this room? And so the first thing, I'll start at the bottom here. The first thing we need to think about is we need to first cross the olfactory epithelium. So these are things that we always, typically would think about: the permeability, the molecular weight, the charge, the lipophilicity. Do we need to use a permeation enhancer or a mucoadhesive to improve transport? But as I alluded to before, there are other challenges that we need to learn more about, and that there is the bulk flow processes that I mentioned. And so if the drug goes through, an example, paracellular transport, it first has to cross the lamina propria, which is rich in blood vessels and lymphatics. So if the drug gets taken up by these blood vessels, it's cleared from the brain. It doesn't even have a chance to enter into the CSF. And then, so when we talked about the lymphatic pathways, And then if we can get it into the subarachnoid space or the olfactory bulb, then we have to think about how that drug is going to be transported throughout the brain. So, I think one of the challenges we have to face here is that, in addition to the typical, epithelial membranes we think about, there are other transport mechanisms at play here. So at Aptar, some of the things that we have tried to do, at least from a delivery standpoint, is to facilitate more deposition in the olfactory region. So we have developed a targeted nasal spray for olfactory delivery. And we've built in some features to help improve the targeting aspect and the alignment of the nasal cavity and the depth of insertion. And just quickly here, you can see on the graph, if we look at the percent deposition in different regions using our AeroNose nasal cast, you can see that, at least from the olfactory standpoint, we can increase deposition by about 40% in the olfactory region compared to a more standard nasal spray platform. And we do also target the turbinate at the same time. So as we noted, it is possible that there is transport going along the olfactory and trigeminal nerves. Other areas in that we're looking at is to try to begin to understand those transport mechanisms. We have a program going on at Macquarie University with YY and Danny to start to look at those cellular mechanisms. So growing, co-culturing epithelial and neuronal cells into a culture transwell, that we can start to understand how the transport, the extent of transport, and then potentially even, put those transwells into a nasal cast using some of the technology that they've already developed. So we're actively working on that program. Also, there are other people that have looked at trying to have a brain on a chip or a nose on a chip. YY has done some of that work, and there are other companies that are doing that as well to try and understand those transport mechanisms. And quickly, transitioning to a few other things before I close the presentation. One of the things that I think has been a potentially groundbreaking change is the self-administration of vaccines. So just recently, the FDA approved FluMist for at-home administration. So FluMist itself was approved in 2003, but what is new is that the nasal vaccine can be shipped to the user, and they could administer the vaccine themselves at home. So it's shipped using a kit, and then the user needs to return the vaccine to be destroyed via another process using a kit. Because one of the challenges is that FluMist is a live attenuated vaccine, so we couldn't have people just tossing their vaccine into the bin. So there needed to be a collection mechanism. So that was actually one of the challenges. So I think this is not yet being sold on the market, but I think it is a really interesting time. It shows the value and the ease of use of nasal administration for yet another. In the hands of the patient for yet another type of therapy. And currently we see there's at least 10, maybe more, nasal vaccines in clinical trials and many more in development in preclinical studies. So hopefully this will incentivize further the development of nasal vaccines. And then finally, we've heard in the last day or two about, training, the importance of using devices from an inhalation standpoint. But the same can be said sometimes for nasal, especially in an emergency situation. You need to act quickly. And so one way to help do that is to have onboarding and training kits. And our Noble, which is an Aptar pharma company, does make training kits, which you can see here on the far left, that mimic the exact feel of what the medication delivery would feel like. And so the graph here shows the Aptar unit dose with the Noble trainer and how it can actually mimic the activation force to help enable that same sensation for when to facilitate training. There are other trainers out there. Not all are meant meet the same feel for the activation force. So just wanted to highlight the value of having a trainer that can actually mimic how the device will be used to help facilitate training for those critical situations. So with that, I'd like to thank you all for your attention and thank you for having me here. I think that the nasal delivery it's an exciting time. Just looking at the growth of the posters and the presentations here, I think that that's evidence of that. It's an exciting time to understand nose to brain delivery. We're seeing it in animals. I'm looking forward to the next decade, or maybe next year if Suzanne's study comes out, that we'll see that transport proven for nose to brain in man. But obviously we need to understand those mechanisms further so we can really optimize drug delivery to the CNS. And as I just mentioned, there's huge opportunities, I think, in the growth for nasal vaccination. And also, always as a reminder, we need to always remember the patient. So with that, I'd like to thank you for your attention. I'd also like to acknowledge the work that we are doing with Wake Forest as well as Macquarie University. So thank you for your time, and I think we have some time for questions. Yeah.
Thank you very much for this excellent presentation, giving us an introduction into nasal delivery. So if there are questions, just show up or come to the microphone. So maybe I'll start with one from Slido. You showed the delivery of insulin to the CNS via intranasal delivery. Do you think that also larger proteins could be delivered to the CNS via nasal delivery?
I do think it's possible. I mean, if you look, viruses and bacteria can enter the nose to brain. So I do think there's room for larger molecules. They may need to be targeted or take advantage of a receptor. So in the nose, there's transferrin receptors, there are insulin receptors, there's GLP-1 receptors. So we may need to focus on a specific mechanism, but I do think larger molecules can enter the brain.
There is a question.
Thanks, Julie. It was a great talk. This is David Morton from Deakin.
Hi, Dave.
We're seeing an increasing number of powder nasal delivery research. I think it's sort of, it's growing recently. Could you comment about the sort of just a general pattern of trend? Do you see that increasing, and what are the sort of what are the influences behind that?
Sure. We do see it increasing, particularly for biologics. It can overcome shelf life issues in that the powder can obviously be stored at room temperature. The nasal powders also have an advantage in that they could deliver a larger payload. So if you're, you don't have the potency or the solubility that you need to achieve the dose, a nasal powder also affords that route. So those are the two main drivers in that sense. And we're also interested to see as to whether or not a nasal powder can actually provide longer retention in the nasal cavity. There's some thoughts that it can. And so because the powder will actually interact with the mucus, the mucin. And so I think that also needs to play out, and that might also, provide a way of having some additional mucoadhesive activities as well. Yes?
So, yeah. Do you see another question? Yeah, okay.
So you mentioned that now we have some vaccines coming up for nasal delivery and that it's live attenuated viruses. How do you think that we can improve these vaccines so that the patient doesn't have to take care of depositing the live attenuated viruses?
I'm sorry, what was the last part of that question?
How we can improve the vaccines so that the patient doesn't have to take care of depositing it in the right way and all that stuff.
Yeah, I think that's going to be the challenge. We've seen, if you look at like recycling programs for devices, they haven't always been successful in meeting those targets. So it's certainly something we're going to have to watch that space. Hopefully, people will follow the instructions and utilize those kits as appropriate. But not all vaccines, I'll say, and we'll probably learn a little bit, not all nasal vaccines are live attenuated. There are many, many other moieties being used for vaccination that are actually a live vaccine.
Thank you very much. I'm sorry I missed the beginning of the presentation, but the penultimate slide showed somebody putting the nasal nozzle into the right nostril using the right hand. We've always been taught that this is an incorrect technique. You should be using the left hand for the right nostril and the right hand for the left nostril. The rationale being is that people do tend to jab the nasal inhaler into their noses, and they score the septum, causing bleeding. And this causes a future aversion to the use of any nasal medication, whereas the lateral nasal wall is far more forgiving. It's also got a thicker mucous membrane.
Yeah. So there definitely have been examples of using that contralateral hand position in the context of nasal steroids, because what actually could happen is if you aim and hit the septum, the steroid can actually cause the nasal bleeding. It's the mechanism that we. No.
It's not the steroid that causes the bleeding. It's the trauma caused by the device itself.
Yeah.
Yes, if you use nasal steroids to the nasal septum over time without traumatizing it, it causes thinning of the nasal mucosa, and that does cause bleeding.
Yeah.
But any nasal insertion of stuff up the nose can cause trauma to the nasal septum, and I think that message needs to be got across.
Yeah, so certainly the nasal sprays have a long history of use over decades, and if you don't mind me answering the question, there are the instructions for use need to be clear for the user. The targeted device that I talked about has a mechanism to align the device in the nostril to prevent targeting either the wall of the nasal cavity. So it is something that we are looking at.
With due respect, people don't tend to read the instructions. They put the
I'm
thing
a
in their
pharmacist,
nose.
sir. I know that that's a challenge.
It's a challenge. And I'm just telling you from experience of seeing people with nasal bleeding post use of a nasal device over many years of practice, that this is a common finding.
Thank you for making us aware of that.
So, as there are no immediate further questions, thank you again.
Thank you.